The presentation that we're going to deliver today is Unlocking Biomolecular Secrets, Comprehensive Biopharmaceutical Characterization Workflows. And I'm really pleased to be here speaking to you for a few reasons. It's my first ever HPLC. I'm honoured to be speaking next to Christine Hoover. And also it's the 50th anniversary of HPLC, so what better meeting to have my first experience here. So without further ado, let's get started. So as part of this presentation, we're going to be looking at some of the analytical challenges that biotherapeutics present and what tools we might need to unlock their secrets. What do we need in our Biopharma toolkit in order to get from vial to file? And finally, we'll take a look at some of the fundamental biopharmaceutical workflows, and I'm going to focus on some peptide mapping and intact, denatured and native data, which is hot off the press from ASMS. So let's start with a question. What are the challenges that biotherapeutics present? Well, the pharmaceutical industry has made a significant transition from the generation of small molecule therapeutics to the generation of biologics. And the reason for this is the specificity of action that these molecules offer and the reduced side effects. And they've really opened the door in treating immune diseases and autoimmune diseases and cancers, which were previously never thought possible. So in terms of analytical complexity, we've also seen a massive increase. So if we think about a traditional small molecule drug, such as aspirin, you've got 180 molecular weight. It's relatively simple to isolate and to detect. A chemotherapeutic agent, such as methotrexate, molecular weight 450, so still pretty small, but its mode of action is a DNA damaging agent. So upon administration, any cells which are dividing are destroyed, and that's why we see such broad-ranging and debilitating side effects when it acts on hair follicle and gut cells, for example. A small protein such as insulin, with a molecular weight of 5,800, was heralded as the first true biotherapeutic after Eli Lilly gained FDA approval in 1982 for biosynthetic insulin. The first monoclonal antibody drugs were licensed around 30 years ago now, and these really offer specificity in action because they're able to bind to and flag disease-causing cells for destruction. And the most recent class of biotherapeutics, known as antibody-drug conjugates or ADCs, offer further advances because here we have the binding of a cytotoxic small molecule with the antibody. So they're able to deliver an intense chemotherapeutic or radiotherapeutic dose of drug to kill that cell. So all of this, to the analyst, presents an increasing level of challenge. Not only have we got increasing molecular weight, with antibodies being around 150,000 Daltons, we've also got a degree of heterogeneity in those molecules that we need to characterize and understand. These inherently complex therapeutics call for the most advanced, fast, and efficient characterization workflows. So, as their name suggests, biotherapeutics are biologically produced, and as with most things in biology, we have heterogeneity in the end product. And it's this that we need to characterize. Now, whilst we expect this heterogeneity, we need to ensure that it's consistent from batch to batch. So this involves controlling the biomanufacturing. So we know that small changes in the culture media, such as pH, temperature, the nutrients involved, can all have an effect on the end molecules that are generated. Very early on in the development of any biotherapeutic, a number of critical quality attributes, or CQAs, are defined, and it's these that we need to build our analytical techniques around. So in order to routinely measure the critical quality attributes which dictate the safety and efficacy of the drug, we need tools such as standardized sample preparation tools, relevant reagents, highly efficient and high resolution chromatographic separation, high resolution mass spectrometry and other sensitive and linear methods of detection, and last but not least, we need intuitive workflow driven software to allow us to delve into that data and really find out the answers we need to speed up the drug production process. So the features that we observe in a protein are a result of its expression, and anything that happens thereafter will influence post-translational modifications. And an example that I like to use to demonstrate this is my very, very old Andrex puppy Teddy. Ben, he's called. And I do still sleep with him every night. He's 16 years my senior, so I'll let you approximate his age. And the reason that he's in such a state is due to a number of things. Is it because of the way he was manufactured? Was it shoddy stitching, cheap stuffing? And we can relate that to a protein. So the main amino acid sequence of the protein is due to the way it was expressed or synthesized. Other things which influence the way that he looks is the way that he's been treated over the years. So I haven't looked after him very well. He's been through more laundry systems and in the Royal Mail system to get him home to me more times than I care to remember. And with a monoclonal antibody, all of the things that we do after it's been synthesized, such as the purification, polishing process, anything that it comes into contact with, that it might react with or bind to, can influence the post-translational modifications that we see. And finally, part of the reason that he looks so bad is because he's aged. He's very old. And the same can be said for proteins. So after they've been synthesized, leaving them for a long period of time at different pH, different temperatures, or even at room temperature on the bench for a long period of time can actually induce some modifications. So some of the most common critical quality attributes that are monitored in a biotherapeutic monoclonal antibody-based drug, as we can see here, are things such as deamidation of asparagine and glutamine residues, oxidation. We need to characterize exactly the position of the disulfide bonds and ensure that this doesn't change. Glycosylation is one of the most heavily regulated areas due to the fact that the glycans which are attached to the protein can dictate its half-life and also how immunogenic the protein is. We also need to look at aggregates, and then when we think about antibody-drug conjugates, we have another level of detail that we need to look into. So the ratio of drug to antibody and any linker molecules which are involved there. It's a real marriage of the small molecule world and the protein world and requires a whole other set of analytical tools. So now let's think about what we need in our Biopharma toolkit in order to characterize these molecules. Well, it helps first of all to think about what an ideal workflow looks like. And I've called this vial to file, but it could quite easily be considered taking a sample through to knowledge. So the idea is that we learn something about that sample, about that protein, in order to make better decisions and modify the process if necessary. So first of all, we have our sample, and that could be a range of things. It could be the intact protein, it could be higher order structures, it could be charge variants, or it could be glycans or peptides or fragments. The first thing we need to do is perform some sort of preparation, whether that's just a simple immunoaffinity cleanup or whether it's digestion into smaller parts. We then need high resolution chromatographic separations and appropriate column chemistries. We need sensitive and linear detectors, and finally we need software to interrogate and feedback. So it's fair to say that chromatography is at the heart of all of these characterization workflows. So it's important that you select the most appropriate UHPLC system as early as possible in the development process to allow you to stay ahead in the rapidly expanding world of biopharma. So let's start with sample prep. So we take our intact antibody, and of course we do perform some analysis at the intact level. We can gain a lot of knowledge. But more often than not, we break the molecule down into its constituent parts to simplify the analysis and to really dig down and get a higher level of detail. So you can use PNGase-F, for example, to release the glycans and look at these separate from the protein. You can also look at the protein after you've cleaved the glycans so that it makes that analysis less complicated. You can cleave just below the hinge region using enzymes such as IDS and papain, and then you can choose whether to reduce the disulfides or not to get reduced subdomains. You could do a partial reduction to look at their light and heavy chains separately. You also need to characterize the higher order structures which are present. So proteins have a propensity to aggregate into dimers, trimers, tetramers, even higher. And it's really important that we characterize this and ensure that the levels of different conjugates are consistent from one batch to the next, because these can be involved in immune response. We do of course look at the protein at the intact level, and that's very useful to get a good overview of the fingerprint of the molecule, so what glycoforms are present and the major modifications. And it's worth mentioning at this point that different methods of detection can be useful for different types of samples. So native mass spectrometry is actually really useful for the analysis of higher order structures and intact proteins, and we'll take a little look at that later on. And finally, peptide mapping. So this is where we take the protein, digest it down into its constituent peptides, and this gives us a level of detail at the amino acid level and the site and the level of the modifications on the protein. So most of the reagents that are required to do all of this can be obtained from our Pierce subsidiary. But I wanted to tell you about one of the new products that we have for generation of peptide mapping data. So these are the Smart Digest kits, and in these kits you get a collection plate and 96 PCR tubes. Now within each one of those tubes is an immobilized, heat-stable resin, immobilized trypsin, heat-stable. So all you have to do is 3 simple steps. There's no need for reduction and no need for alkylation prior to digestion. You simply add your protein and the pre-prepared Smart Digest buffer, which has been optimized for digestion at high temperatures. Add this to the tubes, heat and shake it. Usually about an hour is sufficient for an intact antibody to gain complete digestion. And finally, you might want to do a centrifugation or a solid phase extraction cleanup, just to isolate your peptides for LC-UV or LC-MS analysis. So these kits really offer a reproducible protein digestion, standardized, and you get everything that you need in the box. So from one individual monoclonal antibody sample, you can generate a broad range of substituent samples. Now all of these will lend themselves to slightly different column chemistries for the separation and slightly different detectors. But it's fair to say that if you had the right UHPLC system and the right mass spectrometer, you could analyze all of these with a single system. So when we think about separation, I'm sure you've seen and heard about our latest addition to the UHPLC family at Thermo, and this is the Vanquish UHPLC system. So when we decided to make a new UHPLC, we didn't want to make incremental improvements on an existing platform or add extra bits of technology. We really wanted to go back to the drawing board and think about the latest advances in column chemistry and what our users really want from a system. So we did extensive voice of customer surveys and listened to our customers, and the Vanquish was the end result of that. So you may have heard us talk about the Vanquish being built for biopharma. So the reason that we say that is because biopharmaceuticals are one of the most challenging separations that anybody will have to deal with. So if the Vanquish was custom built, i.e. all the tools and features in there are ideal for proteins, then it's going to be able to address any application area as well. So the Vanquish Horizon is our high-end system, and this operates up to 1500 bar and is a binary pumping system, and it incorporates the latest cutting-edge technology in pump and valve design. Now last year we launched the Flex Quaternary system to the family, and this offers a level of versatility with its quaternary pumping, and it goes up to 1000 bar, and it's great for all standalone or LC-MS biopharmaceutical characterization. The latest addition, which you may have seen, is the Flex binary system, which we've launched here at HPLC this year. And this brings the high throughput, high pressure mixing capabilities to the Flex family. So we really do have the most cutting edge fit for purpose UHPLC. And I wanted to just spend a minute looking a bit more closely at the Vanquish. So aside from its sleek exterior, which a lot of people have likened to be something like you would see NASA produce or something out of Star Wars, if we open the doors and take a look inside, you can see that a lot of thought and consideration has gone into the layout of the system. So there's not tubing hanging out of the sides everywhere. Everything is very easily accessible. There are lights down in the pumping area so that you can see exactly what you're doing. And it makes use of our Viper finger-type fittings throughout, so you don't need to go hunting for the right size spanner whenever you want to make any adjustments. So So a lot of the features that we built in are for simplicity, so tool-free fluidics, maintenance-free valves. It's got a compact footprint, so it can fit into even the most space-limited labs. It's very easy to switch in and out the modules. You don't have to destack to perform any maintenance either. Well, the Vanquish delivers precise flow rates and gradients across the full proportioning scale. It has ultra-low pump pressure ripple, and it incorporates our light pipe flow cell technology for the ultimate sensitivity and signal to noise in the UV detector. We have accurate temperature control, whether that's in the sample manager or in the column compartment. And we also have our patented Smart Inject technology. So this acts to it's sample pre-compression. So once the sample has been taken into the loop, it's brought up to system pressure before we switch the valve. So what this does is it minimizes any pressure shock to the columns and any fragile stationary phases. Yeah, so the Smart Inject really acts to minimize any pressure shock to the columns. And the upshot of this in terms of the data is that we get highly reproducible retention times from run to run. And Frank talked a little bit about that yesterday. And this is really important in biopharma. If you transition from LC-MS to LC-UV, you need to be confident that that retention time is for the same molecule. So versatility, I think everybody wants a system which is versatile. So on the Vanquish we have customizable gradient delay volumes, and there are a range of spin flow mixers that you can swap out with different volumes. Multiple heating modes in the column compartment with still or forced air, and you can also use active or passive eluent preheating and post-column cooling. It has an extremely large sample capacity, so there are 4 sample plates as standard, and if you get the charger that can be 23, which is nearly 9,000 samples depending on the plates that you use. We've got a broad flow rate range and a temperature range, so up to 5 mils a minute on the horizon and up to 8 mils a minute on the flex. And there are 4 different detectors to choose from. It also supports 30 centimeter columns, 2 of those are multiple smaller columns. Now finally, in terms of confidence, which is also important in biopharma, we want to be confident that our sample is safe in that sample manager, that it's not going to degrade over time. We want to be confident and have traceability of where our samples are. So we've got the barcode reader for the plates. We've got dry air sample cooling, so no more misting up of the window on the autosampler. And the whole system is driven by our Chameleon chromatography data system, which is well proven for user simplicity and the range of tools that are on offer. So all of these combined give us a system which is really built for biopharma. The next thing to think about with a UHPLC system is how easy is it going to be to integrate into the lab? So will the users like it? Will it be simple to set up? Can I transfer my methods easily? And can I develop new methods easily on the system? And I think the answer to all of these is yes with the Vanquish. So first of all, we have the Apps Lab library, which is a fully searchable online repository of methods. So in a single click, you can download an e-workflow and load that up onto your system and you're ready to run. Also, the system we have a broad range of columns and sample preparation reagents, and they're all compatible with our Viper finger-tight zero dead volume connections. The system's driven by Chameleon. It's integrated easily with all of our mass spectrometers and others, and it offers seamless method transfer tools. So let's think about the column chemistries that are on offer. Well, we have a broad range of column chemistries for proteins and peptides, as you can see. We also have the MS pack desaltering cartridge, which is great for quick, rapid profiling of intact proteins. We recognize that all proteins are not equal and that monoclonal antibodies require a little bit more time and attention in the separations, and we've developed a line of columns known as the MAD pack to address this. So there are a range of column chemistries for all the different workflows you might need in biopharma here. For glycans, we have the glycan pack and the AccuCore amide hillock columns. So if you want to separate your glycans based on structure, charge, and size, then the mixed mode hillock reverse phase and mixed mode hillock anion exchange columns are great. For those glycans such as those we find in monoclonal antibodies with less or no charges, the amide hillock is great. We also have columns for nucleic acids. The newest columns in our range are the MADPAC-RP and the DNA pack. So the MADPAC-RP is based on a hydrophobic supermacroporous resin, 4 micron particle size, and it's great for high resolution separations of intact proteins and subunits. Has a really long lifetime as well. So as well as the columns, it's worth thinking about the buffers and reagents that we use. And we have these pH gradient buffers that are pre-prepared. You simply need to make a 1 in 10 dilution of them, and you can get reproducible linear pH gradients on the Vanquish system. So it's really easy to set up. You can see 6 overlaid injections of a monoclonal antibody here run on the MADPAC SCX column. And you can see it looks like 1 chromatogram. With the CX1 buffers, it's also really easy to optimize your method, so you can really hone in on the PI range of your protein of interest. So you can see there that we went from 10 minutes down to less than 1 minute for a separation. So as I mentioned, there are 4 detectors. We have UV in the DAD with the light pipe technology. We have the VWD, which is great for peptide mapping applications, a fluorescence detector for glycans, tagged glycans, and also we have the universal charged aerosol detector, which is proving valuable in the analysis of non-labeled glycans. So coupling of the Vanquish with our class-leading Orbitrap mass spectrometry was a bit of a no-brainer. And we have some tools to help with that. So we have a dedicated connection kit so that you can have the shortest flow path from the LC to the MS. You can also position the column oven left or right on the Vanquish to achieve this. The system can be controlled either by Excalibur or by Chromeleon, according to user preference and compliance requirements. And if we take a look at the Q-Exactive family of mass spectrometers on offer, we have the traditional Q-Exactive, which is well renowned as the high resolution mass spectrometry workhorse for all application areas, and particularly it's well known for bottom-up analyses. The Exactive Plus is well renowned for its analysis of native proteins at the intact level, and also we have the Fusion Tribrid mass spectrometers, which excel in all workflows, but they're particularly useful for top-down analyses due to the fact that you have ETD and MS to the N capability there. You may have seen at ASMS a couple of weeks ago that we launched the Q-Exactive Biopharma. So this is a Biopharma option that can be added onto the Q-Exactive Plus or Q-Exactive HF mass spectrometers. And it opens the door to make the Q-Exactive applicable for all biopharma workflows. So within this you have 3 different modes. You have this high mass range mode, so you can acquire data up to 8,000 Daltons, and this is great for native protein analysis. We have a protein mode with optimized pressure regimes to allow you to look at subunits and perform top and middle down analyses. And then finally you can operate the Q-Exactive in its standard mode for peptide mapping. If we think about data processing, we have Biopharma Finder software. So this is a workflow-driven software which is geared towards the analysis of intact proteins and peptide maps in a single package. You can also perform de novo sequencing in there if you have MSMS data. And it really is designed to get the most out of Orbitrap and ion trap data. It also incorporates a protein sequence manager, so you can input into the software all of your protein sequences, the fixed modifications, the site of the disulfides, and you can keep adding to this so you have a library to refer back to for future analyses. Chromeleon is designed really to increase your lab productivity with the use of e-workflows, and it's known for its fast data processing. So you can control all of your labs, so other LC systems, IC, GC, third-party systems, and it also allows you to operate in a GXP compliant manner. So finally, let's just take a look at the biopharma workflows. So we can broadly divide the workflows in biopharma into 5 areas. So peptide mapping to confirm the amino acid sequence and site of different modifications, intact protein and subunit analyses, aggregate screening, charge variants, and glycans. And for each of these, we require a slightly different column chemistry for the separation, and more often than not, a slightly different method of detection. So Because we don't have time to go through all of these, I'm just going to show you some of the latest data we've got for peptide mapping and for intact protein data. So our peptide mapping workflow is really simple. As I've already said, we use the Smart Digest kits to digest our protein, and then we can analyze via LC-UV or LC-MS and process the data in Chameleon and/or Biopharma Finder. So I'm going to show you some data from a digest that was done on rituximab using Smart Digest. So all we do is dilute the protein 1:4 with the buffer in the kit, leave it for an hour at 70 degrees, and we're ready to run. The column that we use for peptide mapping is the Acclaim Vanquish C18120 column, and we analyze this on the Q-Exactive with Biopharma Finder. So here you can see 13 consecutive chromatographic runs of a Smart Digest sample. And you can see here that if it weren't for the zoomed in regions where we can see the most hydrophobic and hydrophilic peptides, there really doesn't look to be much difference in the overlays there. And if we look at the RSDs, the specifications of the Vanquish are 0.15%. You can see it easily surpasses its own specifications, and more often than not, we hit RSDs of less than 0.02 or even 0.01%. So you can really have confidence that you're making the correct assignments every single time. Now we wanted to push the Vanquish system and see what we could really achieve with our peptide maps. So peptide mapping is reputedly quite a long separation process. Gradients of an hour or more are not uncommon. So we took a standard 30-minute gradient, which looks quite nice, and we said, how fast can we push this? So through a combination of increasing the flow rate and shortening the gradient, we were actually able to achieve a really short 5-minute gradient on the Vanquish Horizon system. So this gave a back pressure of 1300 bar. The Vanquish Flex system would easily be able to achieve the 13-minute gradient that you see in the middle there at 850 bar. Now that's great, but when we run a peptide mapping experiment, what we're actually trying to do is to get all of the protein. We want to get 100% sequence coverage. So the question here is, well, we've shortened it up. Have we still got 100% coverage? And when we processed with Biopharma Finder, we found that we did actually have 100% sequence coverage even down at the 5-minute separation. Now, of course, if you want to get that level of detail of the really low level glycoforms and modifications, then you probably won't see all of those in the 5-minute gradient, but we did see all of the amino acid sequence and the major variants. So the next thing, if you speed up your peptide map to 5 minutes, is to think about the detector. So we've now got a really fast separation, 100% coverage, but how fast was the detector able to keep up with that? Were we able to get sufficient scans? Well, when we looked into this, there were 60 peaks in that 5-minute chromatogram, an average peak width at half height of 1.4 seconds. So if we take a look at this peak at 1 minute that's highlighted here, the peak width at half height of that was 0.9 seconds. At Bath it was more like 2.4 seconds. And we used a top 5 DD MS2 method. So what we actually have underneath that peak is 6 full scan MS spectra at 60,000 resolution, and we have 5 times top 5 MS-MS spectra, which totals 31 scans across that very narrow peak. So the upshot of this is that the Q-Exactive is able to scan more than fast enough to be able to profile the peak quantitatively and qualitatively. And if we had co-eluting peptides, we should also be able to identify these with this scan rate. So finally, for intact protein, well, there's lots of different ways we can look at intact proteins. It might be an affinity chromatography cleanup, and we might be checking the titer. Perhaps it's the intact protein and we're just looking at the major glycoforms. Maybe we're using size exclusion to look at aggregates or charge variants. Or for antibody-drug conjugates, we might be using hydrophobic interaction chromatography to look at the ratio of drug to antibody. So HIC is proving itself to be the most efficient method of separation for antibody-drug conjugates, because the more drug molecules that we add to the protein, we increase the hydrophobicity. So we can look at that elution order and say how many drugs are attached. But we've also found that using reverse phase and size exclusion can be great for coupling to mass spectrometry. So what we did was we took an antibody drug conjugate and we ran it on the MABPAC-RP column, so under denaturing conditions, and in parallel we ran it under native conditions by size exclusion chromatography. We used ammonium acetate as the eluent here. So if we take a look at that data, so the data was from trastuzumab emtansine, or Kadcyla, which is a lysine-linked ADC. And we look at the native data. So you can see here that we have the total ion chromatogram, the ADC peak eluting first and the salt later. So you could use a divert valve if you wanted to prevent that going into your mass spectrometer. And if we take a look, you can see that the multiply charged envelope is concentrated over a relatively small mass to charge range, and it's most intense around 6,000 Daltons. Now this data was acquired at 35,000 resolution. So you can see the R value that's annotated on there is actually the measured resolution of the end result. And as we zoom closer in, you get more level of detail, and you can see here that we've got quite good baseline resolution of the different forms of the protein. So in its multiply charged state, each one of these peaks slightly dictates a different glycoform or different antibody drug ratio. Now if we compare this to the denatured reverse phase separation of the antibody, again you can see the ADC eluting nicely there. You can see the mass range immediately is much lower, and that's because the protein is carrying more charges, so it sits at a low mass to charge. And also that multiply charged envelope is spread over a broader area, so we've diluted the concentration or the signal. Again, if we zoom in closer, we can see that the resolution is not quite as good as in the native separation. So the bottom line really is that under native conditions we can get a higher spatial resolution, and if we detect at higher mass to charge and we have baseline resolved peaks, even for overlapping peak patterns, it makes our life simpler in interpreting that data. So that's when native mass spectrometry really comes in, and we can gain increased signal to noise as well. So with that, I hope that I've shown you that the use of the appropriate LC-MS workflows and tools is the key to unlocking the secrets of these really complex molecules, and that our Built for Biopharma solutions are really designed so that you can stay ahead in the evolving biotherapeutic landscape. And a quick acknowledgement before I hand over to Professor Huber, just to say that all of the technologies that you've seen here and the data that I've presented wouldn't have been possible without the work of a lot of these people. So I thank them.